Well, dear colleagues, when we are discussing treatment of glaucoma, especially in case of comorbid conditions, first and foremost, we must understand that this topic is very relative due to the fact that indeed this comorbidity occurs rather often. Well, see here, colleagues, indeed, if a patient has diabetes mellitus, if there is diabetes mellitus, the risk of glaucoma will increase by more than one-third. If the metabolic disease by 50%, and discussing treatment of comorbid conditions, once again, I would like to go back to 3P medicine to talk about personalized medicine, and this was mentioned by the previous speaker. Again, see how many studies there were of different substances due to the glaucoma pathogenesis, different immune, genetic, leukemic, hormonal factors, but these were just separate studies. Right now, there is the era of systemic studies, and it's not fortuitous that the previous speaker was talking about proteomics. We talked about proteomics when proteins are being studied, and we're talking about gene diagnosis and treatment, but I would like to draw your attention to metabolomics, which as of today is called the systemic study of unique chemical fingerprints specific to the processes occurring in living cells. Metabolome is a set of metabolites, molecular weight less than 1500 daltons, which are the end product of metabolism, but they reflect the essence of interaction of genetic factors and the factors of the environment. Naturally, of course, this process has always been the subject of our interest. The history of metabolomics go back to the middle of the last century, such as the studies by Roger Williams, but at that time there were no methods to study metabolites as we have today. Now, when we have such methods, and when we studied the human metabolome, it turned out that it contains more than 100,000 metabolites. These methods, such as gas spectrography, mass spectrometry, nuclear magnetic resonance spectroscopy, and some other methods, have opened new vistas in studying of metabolites simultaneously in one drop of physiological material. As you can see here on this slide, you can see the blood, well, also the optical nerve tissues in case of glaucoma, or such things as different other drops, and we can use some modern methods of septic treatment. Then we choose the sample and compare them with healthy individuals, and then we identify metabolic pathways. We do the enrichment analysis of metabolic pathways, and when we have done this, we can see the predictors or biomarkers, metabolic biomarkers, and we do the validation on animals or in a particular clinic on human patients. So this is the difficult path of metabolome studies. How is it possible? See here, colleagues, as of today, 18 studies have been conducted on metabolomics in glaucoma. In 6 cases, we examined blood. The rest examined aqueous humor, tears, and optic nerve samples. A total of 140 metabolites have been identified. But what is important, the meta-analysis of all studies have been published. For example, here is the recent meta-analysis of these 18 studies. And on the slide, you can see the major metabolites. They are linked to glaucoma. And number one is amino acid metabolism dysfunction, such as arginine. All other metabolites, well, show disorders related to oxidative stress, for example, the calcium cascade. Well, methionine is a precursor of homocysteine, and I would like to draw attention to arginine because arginine is the precursor of nitric oxide. And we've been talking a lot about nitric oxide. And I think about 20 years ago, well, we did not pay attention to this particular substance, but now we know that there are eye drops which regulate the metabolism of nitric oxide. Here are our studies conducted more than 20 years ago when we studied the metabolite of nitric oxide and also the aqueous humor of those patients with glaucoma. At the initial stage, there are a lot of metabolites like this. Well, at the initial stage, there is an increased synthesis of nitric oxide, and this is a dangerous situation. I'm trying to show you this slide, like this slide, okay, because nitric oxide in combination with the onions and radicals form peroxynitrite, leading to the cell apoptosis. But nitric oxide is needed because it basically eases trabecular, helps the fluid outflow from the eye, and strengthens the perfusion of the optical nerve head. This is very important to see here at the advanced stage of glaucoma, especially far advanced glaucoma. The synthesis of nitric oxide is very low, extremely low. There is a double problem. Hypersecretion is bad, and insufficient secretion is also a problem. And at the initial stage of full metabolic changes, we can see those related to arginine, and this should be the focus of our attention in searching further treatment. As a neuroprotector in case of glaucoma, more than 30 years ago, we studied aqueous humor in patients with glaucoma. A lot of studies were done. The oxidation of lipids, free radical, oxidation, and you can see that aqueous humor is very low in case of glaucoma. We showed, the problems related to the transport function of albumin in case of glaucoma. And for that time, we used the advanced technologies such as the probes, but colleagues, we could not have imagined at that time that 30 years later, I mean, not just us, but global science will be able to conduct studies when in the drop of aqueous humor, and that's 50 microliters of humor that we can get during the glaucoma surgery. We can study at the same time hundreds of metabolites, and they have been studied today, 140 major metabolites found in the aqueous humor, and we have the major metabolites identified. Also, their interaction is important and has been studied as is shown on the scheme. It is important from the practical point of view. Here you can see one of the recent publications on this subject. And indeed, when we are aware of such metabolites, when we know what the problem is, we can identify the ways of treatment. And this is one of the studies when there were problems with 31 metabolites. And again, you can see it's not arginine, but it's arnitin. And again, it is related to the nitric oxide and thiamine, vitamin B1. And when the substances were found, we tried to treat the isolated culture of cells, and we conducted experiments on animals. We used the PERG. The effect was assessed by PERG and cell count, and it turned out that indeed these drugs significantly improve the course of neuroinflammation, which was modeled on mice, and they were direct neuroprotectors, having an effect on the synthesis of nitric oxide. I would like also to draw your attention to yet another important publication and to show you that these metabolic studies are very important. They were carried out on a large number of patients and healthy individuals, and there is the so-called biobanks. And this study, which was conducted at Stanford University in the United States, included a large number of people. Well, this blood was kept for 10 years, and then those people, who had the glaucoma, had this blood unfrozen, and studies continued. 599 cases of developed glaucoma were selected and 599 controls. What is important concerning this study is that the data obtained as a result of this study were compared later with similar data obtained by the studies in Great Britain. There were also biobanks there, but they were not retrospective studies. They chose about 2,000 patients with glaucoma and 44,000 controls. And in general, um, uh, 369 metabolites were studied and 18 metaboloid classes using nuclear magnetic resonance spectroscopy. What is important as far as the study is concerned, everything was taken into account, not just age and sex, but also such things as patients' habits, or, for example, how often, how many cups of tea or coffee the patient drinks, say, during the day, whether the patient, uses alcohol or systemic diseases such as diabetes mellitus, hypertension, to mention but a few. What are the drugs used for treatment? Different forms of glaucoma were taken into account. Peripheral defects were taken into account, and so on and so forth. And this study showed that, according to the American Biobank and British Biobank, in case of glaucoma, the lipid exchange is violated. There was an increase of triglycerides in plasma, and the conclusion was, and we thought about it many years ago, the lipid profile is poor in case of glaucoma. Even when we try to treat patients with glaucoma using different drugs, and there were different publications, but they were not conclusive. Everything depended on the design of the study, of course. And right now, the question is, should we administer this particular drug? And you can see at the bottom on the left, there are blue bars. That means what was elevated in the patient. Citrate, pyruvate, lactate were elevated, and that's very important because when we see such a situation, these substances can be considered as neuroprotectors. What we can say concerning the data obtained, it turned out that patients, with paracentral defects, had glaucoma associated with the presence of comorbid systemic pathology. The presence of the metabolic profile were quite often, such as diabetes mellitus, cardiovascular diseases, but carnitine, organic acids, and cholesterol ethers had an inverse association with the development of glaucoma in both the paracentral and peripheral visual field effects. None of the metabolites linked to glaucoma, the patients were divided into groups, had any relation to age or to congenital anamnesis, but there was the link with the body weight. And this study shows that probably in time, it makes sense to conduct new studies about the pathogenesis of the disease, and the format of such studies will change. Here are some other results of the data. Carnitine was low, and that means there is a need to use a low carbohydrate diet and add specific organic acids. Pyruvate, acetate, lactate are anti-risk factors. And here is proof to that. There is the recent study showing that people who had genetic predisposition to glaucoma, but who had these metabolites in blood in elevated concentration, did not develop a glaucoma. So we can consider these substances as direct neuroprotectors. So if you look at the major metabolic pathways in glaucoma, which I have already covered them, arginine, amino acids, lipid metabolism, and now mitochondrial dysfunction. And that's what we have known for a long time. Glaucoma, in its essence, has violations of mitochondrial function, poor mitochondrial function, especially depolarization of mitochondria has always been considered as a predictor, as an early predictor of glaucoma. And now you can see the metabolomic studies have shown that in case of glaucoma, while a syn carnitine level is low, that means that the mitochondrial dysfunction is involved into the epigenesis of glaucoma. Beta oxidation is one of the key aspects of the glaucoma pathophysiology. This is a mitochondrial depolarization, which I have already mentioned, which was solved way back in 2017. And here you can see the drugs used for treatment of glaucoma in terms of mitochondrial dysfunction. In principle, you know all of them. Okay, you can see coenzyme Q10, ketogenic diet, nicotinamide, well, resveratrol, even the drops containing coenzyme Q10 are on the market. They were developed in Italy. Resveratrol is also used to treat the mitochondrial dysfunction. Glaucoma for a long time has been treated by such a drug as tocopherol and others, but I would like to concentrate your attention on nicotinamide because right now all the global attention is focused on this particular drug. A lot of multi-center studies have been conducted here, and nicotinamide, B3 vitamin, and here is the recent publication on this issue. The patient had the metabolic profile studied, and for 2 weeks they were on nicotinamide. And I should say that this drug has side effects. It is hepatotoxic, and here you can see the safety profile, but the treatment was for 2 weeks only. After 2 weeks, the metabolomic profile was studied, and there were positive changes here. Moreover, there were patients with hypertension, with different types of glaucoma, and there was even the moderate involvement of mitochondrial DNA in blood of the patients, but not in case of glaucoma over this short period of treatment. So this question is the subject of study, but if you look at our Russian market of drugs, first and foremost, we're talking about the picamilon, which contains vitamin B3 molecules, as well as gamma aminobutyric acid, which is a neurotransmitter in the CNS. It has nootropic, tranquilizing, antiplatelet, and antioxidant effects, and has an impact on the mitochondrial dysfunction. It can be used as a neuroprotector in case of glaucoma. Recently, we published a review devoted to picamilon. Unfortunately, there haven't been a lot of clinical trials. There was just one article on the subject where patients for 1.5 months were on picamilon, and their vision acuity improved at the end of treatment, and there were also improved trends in the retina condition. Colleagues, the metabolic profile in different forms of ophthalmic pathology was studied. Again, you can see such as AMD, retinopathy. We can have the maps of these metabolomic profiles. We can do the comparison. And if we do our best, as we did it with AI, we found those metabolic pathways which basically allow us to change the course of the disease, such as AMD, glaucoma, diabetic retinopathy, myopia, and arginine, well, plays an important role here. So nitric oxide plays an important role in pathogenesis of all these diseases. That's exactly true. Let's look at the AMD. Well, that's the violation of the lipid metabolism. We've known this for a long time. Now we got proof of it, scientific proof of it. You can see also glucose metabolism is very poor and the carnitine metabolism is very poor. That means mitochondrial dysfunction is involved here in the pathogenesis of this disease as well. And what is even more important is that the metabolites such as glutamine, glutamate, well, like in case of glaucoma, alanine, aspartate, these are metabolic pathways related to the quick progression of AMD, and there are also metabolites which are not subjected to different treatments of glaucoma by specific drugs. Well, diabetes mellitus and glaucoma, what do they have in common? Well, it is difficult to imagine that a long time ago, diabetes mellitus has been considered as the anti-risk factor in case of glaucoma. There were calculators offered by American researchers, and we just used the diabetes mellitus as an anti-risk factor. It was a long time ago. Young people probably don't remember that. Right now, there are data showing that it's quite the opposite, quite vice versa. And you can see a lot of joint metabolic pathways. Well, lipid metabolism disorders also increased NO production and cardiovascular problems. Well, also extracellular matrix accumulation increases, the ocular pressure and also pathological remodeling of the membrane cells. The patient with diabetes will have a faster progression of glaucoma. Also, in case of diabetes, there are also not the paracentral defects, but peripheral, well, defects in case of glaucoma. But what is also interesting is that the drug which is used to treat diabetes mellitus quite often, that's metformin, has the neuroprotective action. Maybe for this reason, when earlier we had the patients with diabetes mellitus, well, glaucoma was not detected quite often in these patients. Well, that was my hypothesis, but a lot of things were discovered due to metabolomic studies. We know that in case of diabetes and glaucoma, the resistance to insulin is elevated because, well, that's important to remember in case of glaucoma. When we try to treat lab animals with hypertension with insulin, it turned out that these animals had the dendrites restored and the pulse to the brain cortex, transfers much faster. You can see insulin and treatment of glaucoma, prostaglandin analogs and treatment of diabetes. When we studied the metabolic profile in case of diabetes, glucosinoids, for example, the level of these prostaglandins was very low, and we started treating lab animals with lantanoprost, and we saw that there was the improvement of the capillary conditions. So this is the global mechanism when everything starts with, and the hypothesis is maybe xalatan can be useful in treatment of retinopathy. This is just a hypothesis, but we know all too well that there are direct neuroprotective conditions of lantanoprost. They were shown in many experiments. 2 years ago, we published the review of neuroprotective properties of xalatan, also for treatment of ischemia and perfusion models, as well as the peroxide oxygenation of lipids linked in particular to hydrogen oxide. And this property of these ganglia cells were demonstrated in case of latanoprost and litimerol. And it was shown that in combination of these drugs, the survival rate of cells is even higher. And that is why, colleagues, when we consider the fixed combinations of latanoprost and timolol, well, such as xalocom, we must think not just about the high hypertensive activity, but also about the direct neuroprotective property of this fixed combination. That is why the British trial showed the data that glaucoma progressed less in patients who were on latanoprost, and that was placebo-controlled.
And finally, we approach another metabolome mechanism, which is a key one in the pathogenesis of neurodegenerative pathology, including glaucoma. This is oxidative stress. So, without diving deep into the mechanism, let me remind you that we studied properly antioxidant activities of topical drugs for topical treatment of glaucoma. And we've shown that the highest one is for dorzolamide. That's why I'm drawing your attention to dorzolamide, which is generic of this treatment that is having very important antioxidant property. It was shown that dorzolamide increases the resistance of trabecular endothelium, and from the fixed combination, the highest oxidant activity was shown in dorzolamide in combination with timolol. That's why dorzotimol can also be considered as a neuroprotector and direct antioxidant, but not just antihypertensive treatment. Also, antioxidant properties were shown in brimonidine because it increases internal antioxidant activity, glutathione levels specifically, and preserve the tissues with the effect of oxidative stress, reducing the level of malonate aldehyde. That's why it was shown that brimonidine preserves the survival of internal levels of the retina, and we know direct neuroprotective properties of brimonidine and brimonox, which is a generic, could be considered today as direct neuroprotector, but not only hypertensive treatment. So in my presentation, I cannot miss retinolamine because we speak today about amino acids. And dear colleagues, let me remind you that this is ultra-short peptide that has just 4 amino acids in its context. But please appreciate, these 4 amino acids, they had a quite comprehensive effect. So they are antioxidant, and they could treat this mitochondrial dysfunction, also improve cell metabolism, and preserve gangliotic cells. So experimental base for retinolamine is very rich, and highly scientific clinical trials are still ongoing on that, and we have follow-ups showing that retinolamine can preserve visual function in glaucoma, and it is used in our country for many years already. So the question is, what is the delivery mechanism of the drugs which are known or will be synthesized based on metabolomical studies? So how they will get to ganglion cells? So that's an interesting question, but to me it's more disputable. I think it should be delivered in the shape of implants, probably in the vitreous body, like neuroprotective factors, like treatment of macular edemas intravitreally, but maybe it would be simpler, and I'm dreaming about it. Hopefully it would be not the drops, but nasal spray, for example, because such approach is also available now. Well, from amnion, we have multipotent cells, predecessors, precursor cells, and they are delivered through the instillation and even nasal spray, and even ischemic disease treatment now, and there were publications like this, and it can be done with nasal spray containing insulin. So, colleagues, here you can appreciate how interesting it is and how many new things we have in modern science. Metabolomic studies open new perspectives in the understanding of the pathogenesis, in the understanding of comorbidities, and all that allows us to monitor and treat patients properly based on principles of 3P medicine. With that, thank you very much for your attention.